My name is Edmond Young. I'm an associate professor in the Department of Mechanical and Industrial Engineering, and I have a cross appointment to the Institute of Biomedical Engineering. The research that I focus on in my lab involves development of a technology called organs on chips, and that involves building tiny little devices and then injecting cells and biomaterials into them to model parts of the human body. Microfluidics is essentially the study of liquid flow in very, very tiny pipes. So think of the pipes and the plumbing inside your home. Instead of the pipes being this big, think of pipes that are the size of the diameter of one of your hairs. And the reason why we do this and the way that we can achieve this is we've used these microengineering methods that have been adopted from the microelectronics industry to create the computer chips in your laptop. And instead of flowing electrons inside wires, we are now essentially flowing liquids inside of these tiny pipes or microchannels. One of the best applications for microfluidics is point-of-care diagnostics. So imagine if you need to do some testing on someone's blood sample or urine sample or saliva sample, the ability to take very, very small quantities of liquid, take that sample, put it into the microfluidic chip, and have that liquid be dispensed and analyzed inside of that chip, all on a tiny little credit card size platform that sits on the palm of your hand. And we can do all of that in a fraction of the time that it would take for a large laboratory inside of a hospital, for example. So organs on chips are effectively, again, these palm-sized microfluidic devices or miniature devices where if you are able to create microchannels and geometries with just the right architecture and the right arrangement, you can deliver the cells, the biomaterials, and some of these other components into the chip and recreate parts of the human body. And basically where microfluidics comes in is inside of the body, it's full of tiny little blood vessels, capillaries, and even in some of the matrix spaces, the cells are often crawling inside these tiny pores and regions. All of that involves tiny flow inside of tiny channels and chambers. So organs on chips and microfluidics, it's actually like a perfect marriage where you use microfluidics as the mechanism to create these organs on chips for biological applications. So regarding the lung on a chip project that my lab is working on, we're working on it in collaboration with Dr. Amy Wong, who is a scientist at Sick Kids Hospital. And one of the big problems that we're facing right now is that when it comes to respiratory infectious diseases, those are actually on the rise. And not just in regards to the recent COVID-19 pandemic, but also in terms of seasonal flus, the RSV virus. It's sort of in mainstream media that we talk about all these infectious diseases. And a lot of the times we're always in need of developing new drugs for these viral infections. But to develop new drugs and better drugs, we need better models in the lab. And that's where microfluidics and organs on chips comes in. We're trying to develop a brand new human stem cell derived organ on chip to model the tiny little air sacs deep in the lung. And those air sacs are the ones that inflate and deflate when you breathe in and out. And the key with these tiny little air sacs, which are called alveoli, is they're lined with these very delicate epithelial cells on one side and endothelial cells on the other, separated by a very, very thin membrane. So to model this accurately, we need organ-on-chip technology, which is where I come in. And Amy Wong, our collaborator, is the expert in stem cell biology. And so both of us sort of teamed up together on this particular project. So in terms of industry applications for lung on a chip specifically, or even organs on chips in general, a lot of it would be for pharmaceutical companies, for example. I think they would be the ones who would be the end users of this technology. I think they see the benefit in terms of testing the drugs, having it way more accurate than before with all the petri dishes and well plates that they typically use. This would be a definite advance in terms of using technology that replicates and mimics the human body in a much more accurate fashion. So in terms of microfluidics research, what sets U of T apart is really the number of talented researchers that we have here on campus. There's many microfluidics researchers that are working on things like point-of-care diagnostics, as well as organs on chips, along with biofabrication and 3D printing. I think there are lots of really great facilities in the infrastructure that's already established here at U of T. One of them would be the Center for Research and Applications in Fluidic Technologies, CRAFT. So a lot of that money has come from this collaborative center between NRC and U of T to establish the center, and allows all the microfluidic researchers to sort of use the equipment and the instruments that have been accrued by craft. And I think that has really been instrumental, one, for training of the students, but two, to actually produce the prototypes that end up being innovative and commercialized down the road. I think the students are actually the engines of the research. They are the people in the lab, at the front lines, at the bench, running the experiments. And sort of as the researcher in the background, we're there to support them and provide them with all the resources and the infrastructure that they need to carry out their research. But I lean on my students a lot because they're the ones with the passion, and they come in and they want to just learn what scientific research is all about. And I feed off of that passion that they show. So research at U of T in one word, either transformative or maybe even game-changing, I would say.